Phytolyts are microscopic minerals made up from silicates, commonly found inside cells of various plant families. Conifers and angiosperms both produce them, and they are most abundant in the family of grasses, to which the cereals belong. Maybe you have the experience of cutting yourself on a grass leaf, and this happened because of high abundance of these tiny glass-like stones inside the cells. And they are analyzed with the biological microscope, and based on their morphological traits, it is possible to distinguish which plant they belong to. Different plant species form phytolyts of different shape and size inside the cells. In other words, they are species specific, like the pollen grains. Here you can see several examples of phytolyts coming from different species. In some species, different phytolyts are formed even in different plant parts, like leaves, stems, or grains, and by very careful analysis, it is possible to even distinguish from which plant part they originate. And they are extremely enduring and can be expected in a wide range of environments, even in extremes where no other plant fossils can be found. Like, for example, they can endure temperatures higher than 300 degrees. They're quite heavy and dense, so they usually get deposited on the same place where the plant dissolved, unlike pollen grains, which are easily transported by air or water. Apart from all of these advantages, there are also some drawbacks in the studies of phytolyts, like the fact that not all plant species produce them, and that they can be quite similar sometimes. Also, they're not very well studied yet, and there are not many atlases and comparative collections to use in their analysis. They can be obtained from small soil samples, like from cores, like I explained for pollen grains, or they can also be extracted from residues on artifacts, pottery, house floors, and so on. So what can be concluded with the help of phytolyts? We can discuss agricultural practices and land use, diets, artifact use, building techniques, plant translocation, and so on. For example, here we can see an amphora sherd originating from Amarna, and here by analyzing the residue, the researchers could say what was transported inside.